Well, thank you very much for this very flattering introduction. I'm impressed by the tight organization this evening. I'm looking forward to the other talks after the first 30 minutes that I'm going to talk about our research. So what I want to do is I'll give you the seminar actually that I gave about a week ago in the United States. It will be hardcore molecular biology.
Excuse me, could you tilt the screen just a little so that it faces the crowd there?
I'd like that
to be. So while we are tilting the screen,
Yes, it's visible.
better? Or maybe all of you should go to this part. So I'll give the talk the way I give it. It's going to be hardcore molecular biology, but have a lot of small animation movies that should help in case you don't understand the genetics, should help you to understand what I'm trying to convey here. So what you saw behind me was the inside of an intestine, of a gut. What you might not know is that the epithelium of the gut of a mammal, mouse or human, is actually the fastest cell-renewing tissue in your body. So every 4 or 5 days, the entire tissue is renewed, presumably from stem cells. And I don't have a pointer here, so this works. Yeah. And the stem cells, so this is a crypt. We started seeing the villi, which are the darmvlokken in Dutch. And it was known for a long time that somewhere near the bottoms of the crypts there were stem cells, and our definition of stem cells is the following: every organ in your body has adult stem cells, and the stem cells, what they can do, they can either constantly or upon injury, they can produce all of the cell types that belong to that particular organ. So embryonic stem cells make an entire body, adult stem cells, like for instance bone marrow stem cells, will make blood cells, but they cannot make other things. We have a secret guest on So what you see here is a maybe can you do that live? Or will all the lights be out? Well, maybe it's fine. So what you see here is one villus in darmvlok with 2 associated crypts, and you can see the movement of the cells. So they are born in the crypts. Yeah, this is better. They're born in the crypts, and then we move up the sides of the villi, and from the bottom of the crypt, these little holes, the tips of the villi, it takes about 4 days, and then they die, they undergo apoptosis, and they end up in the lumen of the gut. And as you sit here, every day you make about 100 grams of these cells, which is the same order of magnitude as the amount of food that you process with your gut. So it's an amazing amount of tissue that's being produced and degraded every day. Now we've been looking for a long time for these stem cells, and in biology it always helps when you can visualize something under microscopes. That's typically what we do. And what you see here on the left is LGR5 is a gene that was not really studied very well. When we looked where LGR5 was expressed, we noted that these brown little things here were cells that we had entirely missed and were seen actually 45 years ago by a French-Canadian researcher who calls them crypt-based columnar cells. Since then, all of this literature is totally forgotten, and because the papers are not online, you cannot find them in PubMed, we and everybody else in the field were not aware that this discovery had been made such a long time ago. In any case, so there were these tiny little cells hiding between these much larger white cells that are called Paneth cells, and I'll say more about these cells a little bit later. So there's 2 cell types at the bottom of the crypts: crypt-based columnar cells and Paneth cells, and these blue cells here are the daughters, we assumed, of those stem cells that very rapidly proliferate. They're called transit amplifying cells, and in that sense, so when a stem cell divides once, that daughter can very rapidly proliferate many times and make many, many cells, but they're short-lived. Now, to test whether these cells were the real stem cells, we felt that we had to prove 2 things. First of all, we wanted to show that these cells stay in the crypt for the lifetime of, in this case, a mouse, because they have to help make tissue for the lifetime of a mouse, so they have to be long-lived. And second, they have to be able to make all of the cell types of the epithelium, about 6 cell types. And if they would do both, we would state that they are the stem cells. Now, to prove this in an experiment, Nick Barker in the lab, using gene knock-in in knock-in mice, homologous recombination, he inserted into the LGR5 locus, and some of you might lose me here, but I'll show a little movie afterwards. He inserted 2 pieces of DNA. The first one encodes green fluorescent protein. So if this works, these small cells should become green. They should emit green light under the microscope. And then separated by an internal ribosome entry site, which allows you to express 2 proteins from one mRNA molecule. There is a complicated fusion protein, the Cre recombinase, which can recognize small pieces of DNA called LOXP sites and recombine them, and I'll show you that again a little bit later. And this Cre recombinase is made inactive by fusing it to the part of the estrogen receptor, that's the ERT2 part of the protein that you can see. And this makes the protein inactive. It sits in the cytoplasm, it cannot go to the DNA. However, if you inject tamoxifen into the mouse, you can transiently allow it to go to the nucleus and then recombine LOXP sites that we built into the nucleus of these mice. And I'll get back to this. If you don't follow me now, maybe follow me a little bit later. These mice actually are now, we made them about 5 years ago. They're now widely present around the world. In many tissues, much to our surprise, we see green cells, rare cells, but as you can see here, there is 3 villi here. These are the crypts. This is actually live tissue, and this is a magnification here. You can now see that the small green cells discovered 45 years ago by Leblanc, they now are emitting green light, and the cells in between those dark holes are the pennant cells. Now, having seen that these small cells, so it works, our strategy, often it doesn't work, you do your best and you do everything you should do, but then you have the mouse and there's no expression. In this case, the cells were green. We assumed that the other protein, the recombinase that is activatable by tamoxifen, is also in the same cells. And we crossed this mouse to a so-called Cre reporter mouse. This mouse has a gene, transgene, that essentially can turn cells blue by lag Z. However, there is a roadblock X here, so the cells are not blue. The roadblock is flanked by the LOXP sites, and Cre can recombine those LOXP sites and in that sense remove the roadblock. And from that moment on, the cell is blue and can never become white again, and when it divides, all the daughter cells will be blue. Now, you might still not follow me entirely, but the outcome, what we hope to do is this. So we have the daughter mouse, we inject low-dose tamoxifen. One of the small cells, about 15 in a crypt, turns blue, genetically blue, because a piece of DNA is removed. When it divides, the daughter cells will be blue. So the first 2 days, we should see transient amplifying cells appearing that very rapidly proliferate. By day 2 or 3, the first cells reach the crypt to villus junction. They differentiate into one of these cell types. And meanwhile, this ribbon will move up further to the tips of the villi. These differentiated cells are now exposed to the lumen of the gut, so they secrete digestive enzymes, they pump nutrients and liquids into these blood vessels and lymph vessels here. And then by day 4 or 5, the first blue cells should start reaching the tips of the villi. Now, if the cell at the bottom that we marked blue on day 0 of our experiment in an adult mouse, it is really a stem cell. This ribbon should persist. So you constantly lose cells at the tips of the ribbon, at the tips of the villi, but the stem cell replenishes it by producing new cells at the bottom of the ribbon. So we can actually show whether the stem cell produces offspring for the lifetime of a mouse. So we inject it when the mouse is 1 month old, we analyze it when the mouse is 2 years old, and if the ribbons are still there, apparently this stem cell lived for the lifetime of a mouse. And in these ribbons, because they come from a single stem cell, we can look whether there are different cell types, and we can state, okay, this particular stem cell can indeed make all of the cell types of our tissue. Now, that turns out to be the case. This is the real data. So here you see a number of villi, about 10 crypts. In 2 of the crypts, we managed to turn one of the stem cells blue with this Cre-ER fusion protein. 5 days later, we have long ribbons, now hundreds of cells produced from this single stem cell here. 60 days later, I told you that the tissue cell renews every 5 days. So by 60 days, we totally turned over the tissue 12 times, but the ribbons are still there. So apparently the stem cells persist. And even 6 months here, and even 2 years, I don't show here, the ribbons are still there. So the stem cell that we mark blue stays there for the lifetime of a mouse. And in one of those ribbons, as you can see here, we see essentially all of the cell types of the epithelium. So the cell is long-lived, it's multipotent, and therefore is the stem cell of this tissue. And this is actually a graph that Leblanc made 40 years ago, where he predicted that this cell would be the stem cell, and through a number of intermediate steps would make all the cell types of the epithelium. So now we felt, I mean, there's still people in the field that don't believe this is part of science, but we felt that the Blond's predictions was actually true. So the crypt-based polymer cells are the stem cells of the small intestine. And we have these mice where the small cells emit green light. And you can see here it's quite easy to take an intestine out of a mouse, and then under EDTA, when you chelate calcium, the crypts will flow out, will release themselves from the insides of the gut, and you can make crypt preparations. And then using, for instance, trypsin, you can take this crypt apart in individual cells, run them over a FACS, a flow sorter. You're probably familiar with cell sorting. And then, so on the y-axis you get intensity of green light. So essentially the machine makes little droplets, and every droplet has a single cell. And while the droplet falls, it passes through a laser, and there are detectors around it. And as soon as there's a green cell in the droplet, there's a charge on that droplet, and it can be pushed into one tube or into another one. So you can quite easily sort bright LG5 cells as real stem cells from the first generation daughters and from the second generation daughter cells. And these are the bulk of the proliferative cells that are not green, so they're not the stem cells. Now here you see a preparation of pure stem cells, which we essentially get in a few hours out of a mouse. We've done lots of stuff with these stem cells, but the most interesting thing that we did was done by a Japanese gastroenterologist in the lab, Toshi Sato, who's now back in Tokyo to start his own lab. I should say that at that time, this is about 3 years ago, it was believed that it's impossible to take cells from any part of your body, stick them into a culture, and have these cells grow like they would normally proliferate in your body. So there was a dogma that somatic adult cells may be divided a few times, but after 1 or 2 weeks, they will go into senescence or into crisis, everything will die, and if you then wait a long time, sometimes the cell starts growing again, but that cell then typically has transformations, has mutations in its genome. However, Toshi decided to try to grow these stem cells, knowing that they grow very rapidly in the normal gut. They proliferate very fast, they proliferate every day. And we had a good guess about what they needed in terms of growth factors. A Wnt signal, I won't go into too much detail, epithelial growth factor, blocking DMP signaling, and then we did this not on plastic. Often people grow cells in petri dishes, but rather we grow it in Matrix gel, which is a mix of collagen and laminin. And this actually makes a 3D gel, and individual cells can sit surrounded by the gel as if they are in a normal tissue. Now, if you do this with a single stem cell, so if you take a crypt, this crypt will immediately start growing, and we eventually get them growing for 2 years so far. When we take a single stem cell out of the sorter and we stick them in this cocktail, now it becomes a little bit more complicated, but about 1 to 2% of those cells, if we have one cell in a well of a 96-well plate, it will grow out, it will make a very chaotic structure, but then when we break it up every week, we get these much more regular structures. As you can see here, this is actually from human intestine now, and Toshi's Japanese artist's impression is here. What he thinks he sees is what he calls a mini gut. So there is like a normal gut, there is a lumen. There are large numbers of crypts that surround this lumen. These are the rapidly proliferating daughters. The green cells are the stem cells. The cells in between are the pendant cells. And this region here has the differentiated cells. So in essence, all of the cell types are here. Even the kinetics from going from a stem cell, proliferate 2 days, differentiate out, live for another 2 days, and then die and end up in the lumen of the normal gut is exactly recapitulated here. This was a great surprise, that a single stem cell, without any cues other than a few growth factors, can essentially rebuild the normal structure that you have in your body as you sit here. And also from one stem cell, you can make billions of these. If you just culture long enough, all these green cells that you can see here in real life, they are all stem cells, and they're made from one stem cell.